An EEG measures brain activity. Here's how it works. Your brain is made up of billions of neurons, all linked in special ways to achieve its various functions. At some linkage points, we have a neuronal junction, also known as synapse. Looking at one of these connections, we can label the two neurons the presynaptic neuron and the postsynaptic neuron. Alright, so during synaptic transmission, the presynaptic neuron releases neurotransmitters into the postsynaptic neuron. Depending on which neurotransmitter is received, the postsynaptic neuron will either excite or inhibit its charge. In reality, there are many presynaptic neurons all connected to one postsynaptic neuron, so the excitatory or inhibitory response is dictated by the sum of all the neurotransmitters present. Wow, so here's an example. 3 presynaptic neurons are all signaled to release the same neurotransmitter into the postsynaptic neuron. Because these specific neurotransmitters cause a positive charge in the postsynaptic neuron, we consider it an excitatory response, also known as EPSP. On the other hand, if these 3 presynaptic neurons were to all release some different neurotransmitter that inhibits the charge of the postsynaptic neuron, we would call it an IPSP. So here's a quick review. There are 2 types of neurons here. The emitters, presynaptic neurons, and the receiver, postsynaptic neuron. Presynaptic neurons send signals in the form of neurotransmitters to the postsynaptic neurons. Depending on these transmitters, the neuron becomes excited or inhibited. Alright, so now that we understand synaptic transmission at a very basic level, we can learn how to read these signals. The neurons we can read the synaptic transmission from are pyramidal neurons. The only thing necessary to know about pyramidal neurons for this application is that they are uniformly positioned perpendicular to the outside of your brain, aka cortical surface. Depending on the signal received, IPSP or EPSP, they create extracellular imbalances in charge. For example, if a neuron has an IPSP, the intercellular charge will become negatively charged, and the extracellular charge will become relatively positive. Now, because there is a charge imbalance outside of the cell, the extracellular charges will move upwards, to the more negative region in this case, to balance the charge. Congrats, we just found voltages in our brains. Each synaptic transmission only causes a small voltage bump, but because they're happening constantly, the voltage looks very sporadic. We can measure this voltage by putting an electrode on our scalp on the region of neurons that we want to read. Researchers have found that the earlobe is a good spot to reference one electrode's voltage off of. Because the entire body's charge is constantly fluctuating from various factors, we want to eliminate all the electrical noise that this would cause in our reading. Think of it this way. Let's say the body's charge is constantly going up and down like this. If we read the voltage off the earlobe, it will represent the charge of the body. Now, when we measure the voltage of the electrode on the scalp, it will have the same pattern but with small differences from neuron activity. We use a differential amplifier to subtract our reference voltage from our scalp voltage to get a clear signal, and then we amplify it. The resulting signal is our EEG reading. So, let's recap. During synaptic transmission, neurotransmitters cause reactions, which in turn cause charge imbalances outside of the neuron. This creates voltages which can be read from the scalp. We compare these scalp voltages to earlobe voltages to reduce noise. Then we amplify the final signal.